Hi, this is Anuj Kalia in the student section 601 of the Genetics 3112 lab. In the fall 2021 Genetics 3112 lab, we were tasked with understanding the environmental effects on sourdough starters. What is a sourdough starter? A sourdough starter is a mixture of flour and water cultured with various microbes. The sourdough starter is used to cultivate yeast in order to cause bread to rise. The sourdough starter possesses a stable culture of bacteria and yeast. The bacteria the culture possesses are a family of Lactobacilliaceae. The Lactobacilliaceae are very unique. This family of bacteria is both homofermentative and heterofermentative. And what that means is that the bacteria that are homofermentative can ferment glucose into lactic acid using a very simple fermentative pathway, whereas heterofermentative bacteria ferment glucose into lactic acid, ethanol, acetic acid, and CO2 through various different pathways. The experiments were run on various cultures to determine whether or not certain environmental conditions would cause changes in microbial development. The primary environmental change of note was the presence of NaCl, table salt, in the sourdough starter cultures. The students who performed the experiment in person were tasked with determining the volume of the sourdough starters after being exposed to different environments. This was whether or not they were in the presence of sodium, whereas the online students who participated in the online portion were tasked with determining what species of bacteria and yeast were present within the sourdough starters when exposed to different environments. So in order to understand how the data was obtained, it is important to understand the technologies that were being used to obtain this data. The metagenomic data, which was obtained by the in-person students, was obtained through polymerase chain reactions and gel electrophoresis. Polymerase chain reactions, or PCR, are very important to eventually sequence DNA. This is done by taking a single strand of DNA and continuously replicating it until it becomes extremely visible. The DNA for the organisms that were obtained were based with Illumina DNA sequencing technology. The metagenomic assembly was obtained through Kaiju metagenomic taxonomy reads, and the phylogenomic tree was generated by the K-base species tree. All of these online technologies were done through the K-base platform. So for the first week of experimental data, we looked at the control of the original sourdough starters. And as one can note, the Klebsiella pneumoniae and the Lactobacillus plantarum were two very prominent microbes in the control sample. There were also different bacteria such as Staphylococcus, there was E. coli, and different types of lactobacillus. As the week went on, the control microbial community retained a similar character, although the Klebsiella pneumoniae went lower into the dominance levels, whereas the Klebsiella pneumoniae remained pretty dominant throughout the salt-exposed microbial community. A primary difference is that the Lactobacillus plantarum in the control microbial community did not show up. It was much less dominant than the other bacterias, whereas in the salt-exposed microbial community, it did retain some character, although it did lower in its coverage significantly. So after 2 weeks, the control microbial community and salt-exposed microbial community displayed very similar character. The Lactobacillus plantarum was by far the most dominant microbe, similar to the salt-exposed microbial community. And just as well as the in-person data showed that there was not a significant change amongst the size of the microbial communities. The phylogenomic tree shows that the reads, the SRR13355277 and the SRR13355283 reads are both the control and the salt communities respectively, and they showed a very similar genomic similarity to Lactobacillus buchneri. Now, Lactobacillus buchneri is a known heterofermentative bacteria. So in conclusion, after 4 weeks of observation, it was determined that the microbial community remains relatively similar regardless of exposure to NaCl. This has been determined due to a near identical microbial makeup in the culture even after exposure, as well as the in-person data showing no significant changes in size due to exposure. And by mapping metabolic pathways and observing the phylogenomic tree, it has been determined that the sample provided undergoes heterofermentative processes. This is also justified by the primary presence of Lactobacillus plantarum, a known heterofermentative bacteria. The reads were phylogenetically similar to Lactobacillus buchneri and also heterofermentative bacteria. Understanding the effects of certain environments on microbial communities may provide useful insights into the types of conditions needed for certain microbes to flourish. This is important in being able to create certain conditions which may be antibiotic for malignant bacteria and probiotic for helpful bacteria. There are plenty of different factors which can be tested in a similar fashion. One could test the value of temperature, salinity, acidity, etc. to determine whether or not changes in microbial communities occur.